Hey, this is Jonathan from The Body of Evidence. Today I want to talk about the Ice Bucket Challenge. Very recently there was an update in the story of the Ice Bucket Challenge, and I want to confront it because it's a great example of how the media overhypes a scientific discovery. So you may remember the Ice Bucket Challenge. It started in 2014. The idea, it just went viral, and it was this viral charity drive where basically people were dumping buckets of ice cold water on their heads to raise awareness and raise money for research into ALS, or amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. It's what the amazing Professor Stephen Hawking has. Loads of people participated in this. They raised a tremendous amount of money for the American ALS Foundation and a few other satellite organizations, and this money was funneled into research into ALS. Now, at the time, I was a little bit skeptical, but well, not skeptical, but I had some issues with the Ice Bucket Challenge. If you want to know more about this, you can check out a former podcast of mine, Within Reason. It's the episode in which I interviewed Dr. Jill Tarter from SETI. We had a discussion about the Ice Bucket Challenge in the context of effective altruism, which is basically that if you want to affect the greatest number of human lives with the money that you have, donating to research into a very, very rare disease, especially if you don't have any personal connection to this disease, is maybe not the smartest thing to do. Earlier this week, the media were trumpeting loud and clear this discovery in ALS research that was funded by the Ice Bucket Challenge. And the word breakthrough was used a lot. I have here a piece from The Guardian in the United Kingdom that was particularly bizarre in the sort of the slant that it had. Here's a headline: "Remember the Ice Bucket Challenge? It just funded an ALS breakthrough." So the piece is by Nikki Wolf, and it starts with the following paragraph. "It is often easy to dismiss viral charity campaigns as slacktivism, which lacks in real-world impact, but a breakthrough discovery bankrolled by 2014's ALS Ice Bucket Challenge may give the lie to that cynicism." Thank goodness for journalistic neutrality. It goes on to say the following. "The project's researchers announced on Monday that they have identified a new gene associated with the disease, which experts say could lead to new treatment possibilities." And this was, fairly representative of what a lot of media went for. This idea that there was a breakthrough, they discovered a new gene that was associated with ALS, and this could lead to new treatment possibilities. Now, this has been smeared with a lot of hype, and let's try and get rid of that hype. We're going to go upstream the hype ladder and go back up to the press release by the American ALS Association, which funded this research in part and which got a lot of money from the Ice Bucket Challenge. And then we're going to go from that to the original article to see what exactly was discovered, if anything. So the press release I found to be actually pretty good, but it says, ALS Ice Bucket Challenge donations lead to significant gene discovery, that it identified a new ALS gene that now ranks among the most common genes that contribute to the disease. But it also goes on to make some interesting points, which is what percentage of ALS cases are familial, how many are sporadic, and I'll get back to this in a few seconds. It also talks about, what does it talk about? Talk about the fact that protein work needs to be done, and I will mention this at the very end as well. What's interesting is that they end by talking about this new fundraising campaign that is about to start in August called Every Drop Adds Up. So obviously, for them, this is a great opportunity to tell the people who participated 2 years ago, to say, look, your money made a difference. Now we need your money again. Please give back to us. Maybe we'll find a new gene. Who knows? So that was a press release. Now let's go back up to the paper itself. The paper was published in Nature Genetics, which is a very, very good journal, very well regarded. The title is NEC1 Variants Confer Susceptibility to Amyotrophic Lateral Sclerosis. NEC1 is the name of the gene in question. So what did these people do? They did whole exome sequencing. And I have to explain a little bit exactly what this is. So the purpose of your DNA, for the most part, is to code for a protein. And the protein is the effector molecule in the cell. The protein will do stuff. When you have a genetic condition, when you have an inherited disease, what happens is that there's a mutation in the gene that will be transcribed and then translated into a protein. When that mutation is there, the mutation imparts a mistake in the protein. And so the protein will not be the same as it would be normally. That protein will be altered, and that alteration may mean that the protein is misshapen. And if it's misshapen, it may not be able to bind what it's supposed to bind. Proteins work a lot like Legos in a sense, but very specific Legos. And so a lot of these proteins have to bind to each other in specific ways using deeply buried pockets and all this. And so these are called binding sites. And if these are altered because of upstream a mutation in the gene, that's a problem. The protein can't do its job. So in the case of ALS, what they're looking for are these mutations, right, that will cause a protein that malfunctions and thus will cause the disease. So they did a whole exome sequencing. What is exome? Well, the exome is the totality of the parts of the genes that end up in the protein. So they did a whole exome sequencing. So they read every base, every letter that would eventually become a protein. And they were looking for variants. Now, variants is sort of the neutral term when you're not sure if it's a mutation, which is rare, or a polymorphism, which is less rare and may not have any sort of pathological impact. So variant is what they were looking for. It's just this neutral alteration in the DNA. We're not quite sure what it does, but it's a variant. They did a whole exome sequencing on over 1,000 familial cases of ALS and over 7,000 controls. Now, what do we mean by familial cases? Well, it turns out that about 10% of ALS cases are familial, which means that they have a family history. There's a known mutation that is present in the germline that gets passed down from generation to generation. Whereas the sporadic cases don't have that. It seems like the disease just magically appeared in an individual with no known family history. This situation of 90% sporadic cases, 10% familial cases is pretty much what we see in cancer as well. So what the researchers found using this whole exome sequencing is they found in this gene, NEC1, certain variants that were associated with a risk of having familial ALS. These variants were called loss of function variants because they were predicted to result in a protein that would lose its function. As I was mentioning before, if like the binding site, for instance, is changed, the protein doesn't do its job. So it loses its function. Now, this loss of function is inferred. It has not been proven yet, and that's a very important point, something that the media, certainly The Guardian, failed to point out, which is that you can infer loss of function based on the sequence of the DNA. So we know exactly how the DNA is read to make a protein. So we can infer what the impact of a variant in the DNA would be on the protein, but you never know for sure, and you need to actually test it out in a protein, in an actual protein. So this hasn't been done, but these were inferred to be loss of function variants, which is very interesting. The researchers then did a whole genome sequencing, which is similar to whole exome sequencing, in 4 inbred Dutch patients with ALS. And they found, among other things, a different type of variant also in the gene NEC1. This was a very particular, what we call a point mutation. They then looked for these 2 types of variants, a loss of function variant and the one they found in the Dutch patients. They looked for those in thousands of cases of ALS that they had in the bank. And this was their conclusion. They found that these variants were present in 3% of European and European American ALS cases. So if we do the breakdown to be fair to what they discovered, we can see here that these loss of function variants that I was mentioning before were found in 1.2% of familial ALS cases, 1% of sporadic ALS cases, but also 0.17% of controls. The mutation that was found in the Dutch patients, which is an arginine to a histidine change, was found in 1.7% of familial cases, 1.6% of sporadic cases, 0.69% of controls. And they also found some variants of unknown significance at the moment that were also in the gene NEC1, and those were found in 1.8% of familial cases, 1.3% of sporadic cases, 1.2% of controls. Now, to give you an idea, this 3% figure, it's not a lot of cases. However, the previously known risk variants by order of how common they were, we have C9orf72, which is present in fewer than 10% of ALS cases, and the next one down is SOD1, which is present in fewer than 2% of cases. So still, it is a fairly important contributor to risk association to ALS, this 3% figure. But of course, in the eyes, if you don't know anything about genetics, if you don't know anything about the disease, when this is being reported in the media as this is a major gene that has an impact on ALS, you would think that the majority of people with ALS have a mutation in this gene, and that is not what the paper has shown. A quick note about penetrance. As you could see in the table before, some of the controls, so people who don't have the disease, also have these variants. What does that mean? Does this mean it's all BS? Well, no, because there's an extra layer of complexity to the whole thing, which is the concept of genetic penetrance. And I'm not going to go into details, but the basic idea is that just because you have a particular variant in a gene that is known to cause a disease doesn't mean you will have the disease. It also depends on how penetrant this mutation is. So a mutation that is 50% penetrant means that half of the people who have it will develop the disease. So it's not sufficient to have the mutation. There are other factors at play that will influence whether or not this mutation will have an impact on your health. There's an interesting sentence buried in that paper in Nature Genetics, which is the following, quote, "NEC1 has been previously described as a candidate gene for ALS," end quote. Wait a second. Wait, what? Okay, so the newspapers have been saying that this is a discovery of a new gene associated with ALS. That is not quite true. It is more of a confirmation of a gene that we thought was involved in the disease before. And the authors point to two papers that before them had shown a potential association between NEC1 and ALS. Now, the big question is, of course, was this research, were those two papers funded by the Ice Bucket Challenge? This is not an easy question to answer because the money went to a particular association and others as well, and so it's not quite certain who got funded with that money. But if we look at the first of these two papers, the majority of their funding came from a company called Biogen. But some of the researchers were part of this consortium, and the consortium was funded in part by the American ALS Association. The second paper that showed this association between NEC1 and ALS does not appear to have been funded by the Ice Bucket Challenge, but rather by German and Swedish sources. The last thing that I want to bring up is Health News Review, which is a fantastic website that, again, earlier this week, essentially debunked the hype that the media smothered the original paper with. Health News Review is a fantastic website that regularly will take a hard look at what the media says about health and how wrong they are. And they did a very good job of debunking how the media was reporting this new discovery. And I want to read from one of the two experts that they called upon to comment on this story, and that's Dr. Vinay Prasad. He wrote, "This is no breakthrough and another sad and embarrassing example of academics and the media trumpeting very preliminary basic science work far beyond what they ought to. Many, many, many genes have been implicated like this in many, many, many diseases, and we still have not seen any novel therapies making any measurable impact even after many, many years." And what's particularly sad is that the article at Health News Review goes on to cite some tweets that came out in the wake of the media talking about this breakthrough. And one of the tweets said, "Happy this worked, just wish it had back in 1998 when my dad died from ALS." Another one said, "Now there's a cure for it." And this is the problem. This is the problem with the media overhyping these things. People don't understand what breakthrough means. In fact, this was not a breakthrough. This was confirmation of something, of an association that had been shown before. It is good work, like it is solid work, and I am not at all criticizing the authors on this one. The researchers did a very good job. This is a good, this is a solid step forward at the molecular level. We've identified a new genes that is probably associated with ALS, but that's just the beginning of the road. And we don't know for sure that these mutations are causing a bad protein. This is the next step that needs to be done. They need to actually create this protein and see whether or not it does what it's supposed to do. And even when they do this, even if they do prove that, there's no cure. Like this will not, the step after that is not, and here's the treatment. We have known about which gene and which mutations are involved in diseases for a long time. I mean, if you look at BRCA1, BRCA2 mutations that are found in familial cases of breast cancer, we've known about this for a few decades, and there is no treatment for that yet. Now, obviously, further down the road, this may be possible. Of course, if you're following the whole CRISPR saga, this may be where things are headed. There are huge ethical concerns about, gene editing, especially in the germline, which I'm not going to get into right now. But we cannot say that identifying a gene that, when mutated, causes a certain percentage of disease cases, that this will lead to a treatment. That is just not true. And what pisses me the most in this is that if the media keeps on trumpeting this hype and just hyping all of this up whenever they're reporting on health issues, the people who are really concerned about this, the people who have ALS, the people who have cancer, who are reading the news and getting their hopes up each time they hear the word breakthrough, when these breakthroughs fail to materialize concretely for them, i.e. treatment options, they will stop listening to the media. The media will lose the respectability that they have and the credibility that they have. And that would be sad, because who will be left to report objectively on scientific issues?